Hello students, today I am going to speak about the unit 3rd of biochemistry which having the topic beta oxidation of saturated fatty acids and formation of ketone bodies. So, we will discuss it previously about the beta oxidation. From starting of the beta oxidation of fatty acid, we just start with the introduction what is lipid and how they react to form the beta oxidation. Lipids, what are lipids? Lipids are generally, we all knows that lipids are indispersible for cell structures and its function. We all knows that lipids are insoluble in water and they are showing various important functions like cell function and integrity of cell, and they are due to their hydrophobic and non-polar nature. These lipids differ in from rest of the body compounds, and they are also helpful in the formation of cell membrane, and they are unique in their action. What is beta oxidation of saturated fatty acid? What is the mean of saturated here? Saturated mean there is no presence of, there is absence of double bond, and all the carbon present in the fatty acids are saturated. There are no need of any type of double bond in this their structure. So, they are saturated fatty acid, and unsaturated fatty acids had double bond in their structure. So, what is beta oxidation? Why we are knowing the name of unsaturated fatty acid oxidation is beta? Here we have the oxidation of fatty acid in beta position. In beta position of any fatty acid, there is oxidation of fatty acid at beta carbon is known as beta oxidation, and there are so many oxidation also present here like alpha oxidation and gamma oxidation. But in our syllabus, in the syllabus of biochemistry, we are here for the knowing about the beta oxidation. It is in our syllabus of AKTU. So, we are discuss here about the beta oxidation only. So, beta oxidation is oxidation of fatty acid at beta carbon atom or beta position. So, this result is the sequential removal of, they are having the sequential removal of 2 carbon fragment in acetyl coenzyme A. So, what is beta oxidation? Beta oxidation of fatty acid basically involve 3 different stages here. The first one is activation of fatty acid occurring in the cytosol, and the second one is the transport of fatty acid into mitochondria, and the third one is beta oxidation properly in the mitochondrial matrix. The fatty acids which are oxidized here, which are present in the mostly tissues in the body. So, there are so many organs in our body like brain, erythrocytes and adrenal medulla. They cannot utilize fatty acids for energy impairment. Besides these brain, erythrocytes and adrenal medulla, all the other organs utilize these type of fatty acid as they oxidize here for the formation of energy. So, these energies utilized by all the organ or the organs in the body other than brain, erythrocytes and adrenal medulla. So, firstly we have started with the fatty acid oxidation here, fatty acid activation here. How to we are required for the activation of fatty acid? Why we are have necessity for the activation of fatty acid? Because fatty acid is generally present in the body in normal form, and when we have to start the oxidation of fatty acid, we have just start with the activation of that particular fatty acid who's to be oxidized. So, fatty acid are activated in the acyl coenzyme A. Why? Thiokinase or acyl coenzyme A synthase enzyme. This reaction is important, and this reaction occurs in 2 different steps, and these require ATP coenzyme A and magnesium ions also. So, fatty acid which are reacted with the ATP here to form acyl adenylate, which then combine with coenzyme A to form acyl coenzyme A. So, in the first step we have just activate the fatty acid with the help of coenzyme A, ATP and magnesium ion. So, these fatty acids react with the ATPs to activate itself and form acyl adenylate, which then combine to coenzyme A to firstly produce acyl coenzyme A. This acyl coenzyme A again reacted with other compound like the in this activation stage the 2 energy phosphates are utilized here, and here the ATP is converted into the pyrophosphate, which is also work for the this enzyme inorganic pyrophosphate now hydrolyze pyrophosphate to phosphate here, and this ATP is now converted into the pyrophosphate, and this pyrophosphate is hydrolyzes to form the phosphate here, and the intermediate elimination of pyrophosphate made this reaction totally irreversible, because whenever there is elimination of pyrophosphate from this reaction, there is no again combination of this pyrophosphate to this reaction. So, that is why there is no reversible reaction occur here. So, there is no need of pyrophosphate, and when pyrophosphate is removed from the reaction, that is why this reaction is irreversible. So, this reaction is only reversible, this is not reversed, and there is removal of pyrophosphate bond here. So, what is fatty acid activation? Fatty acid activation is conversion of fatty acid to acyl coenzyme A. How this acyl coenzyme A is formed? When fatty acid is reacted with the thiokinase enzyme here in presence of ATP to form pyrophosphate hydrolyze enzyme here, and this pyrophosphate is when converted into the 2 pi bond here, 2 pyrophosphate bond here, and there is formation of acyl adenylate here, and this acyl adenylate is when reacted with the coenzyme A to form adenyl monophosphate, and this adenyl monophosphate is when removed from the reaction, and there is formation of one coenzyme A here, and this adenylate is next converted into the acyl coenzyme A. So, in this reaction there is removal of this pyrophosphate bond here. So, this removal of this pyrophosphate bond is responsible for the irreversibility of this reaction. So, this reaction is not reversible due to removal of this pyrophosphate bond. So, this acyl coenzyme A is the last point here. We have cleared that fatty acid activation occurs in the formation of acyl coenzyme A. The next step after this activation of fatty acid is the transport of acyl coenzyme A into mitochondria. Here transport of acyl coenzyme A into mitochondria is required for the, because the main reason for the transport of acyl coenzyme A into mitochondria is main reason because inner mitochondrial matrix is impermeable to fatty acid. Fatty acids are not directly transfer into the mitochondria. So, there is a specialized carnitine carrier system which is also known as carnitine shuttle is required here for the transport of activated fatty acid here from the cytosol to mitochondria. This occurs in 4 different steps. This occurs in 4 different steps. The first step is due to the presence of acyl group here. So, this acyl group of acyl coenzyme A is transferred to carnitine here. What is carnitine here? Carnitine is a beta hydroxy trimethyl aminobutyrate here. This beta hydroxy 4 trimethyl aminobutyrate is catalyzed by carnitine acyl transferase first, or which is present in the outer surface of inner mitochondrial membrane. So, in this transport of acyl coenzyme A into mitochondria is basically dependent on the carnitine here. This carnitine carrier system is required for the transport of acyl group of acyl coenzyme A to from the cytosol to inner mitochondrial matrix. So, this acyl group acyl carnitine is transported across the membrane by the mitochondrial matrix in a specific carrier system. This is a specific carrier protein, these are known as carnitine, and this carnityl acyl transferase second which is present in the inner mitochondrial endothelial membrane which convert acyl carnitine to return to its previous position, which is known as acyl coenzyme A. The carnitine released here, which is returned to cytosol for its reuse. So, there is a use of carnitine here for the transport of acyl group of this fatty acid from the cytosol to mitochondria, because mitochondria is impermeable for fatty acid here. So, that is why we use carnitine as an carrier protein for the transport of free fatty acid or acyl group of fatty acid, activated fatty acid to from cytosol to mitochondrial or inner mitochondrial matrix. This is also cleared by this step also, because there is long chain fatty acid, long chain acyl coenzyme A, which is transported from the cytosol to inner mitochondrial membrane by special transport system, which is also I have described that this is known as carnitine shuttle or carnitine protein transport system, which is helpful for the transport of acyl group of activated fatty acid from cytosol to mitochondrial matrix. So, this is the general this is the structure of carnitine. Beta hydroxy trimethyl trimethyl aminobutyrate, which is having the structure here, and this is the carnitine. And when this fatty acid, activated fatty acid, is when reacted with the acyl transferase first, and this is converted into two part, fatty acid or acyl group, and the other part is coenzyme A, which is freely transported from the cytosol to inner mitochondrial matrix. But fatty acid is reacted with the carnitine here to form fatty acid carnitine complex or fatty acid carnitine shuttle, so that this free activated fatty acid is transported through cytosol to inner mitochondrial matrix with the help of enzyme translocase. And this fatty acid carnitine is when transported into the inner mitochondrial matrix, this carnitine is free from this complex here, and the freely transported coenzyme A is again reacted with this fatty acid to form fatty acid acyl coenzyme A or acyl coenzyme A, and this carnitine is again reacted back to the cytosol for its reuse, and this acyl transferase second is responsible for the formation of again free fatty acid with its coenzyme A. The third step in this reaction is beta oxidation proper. In this beta oxidation proper, when activated fatty acid occur and the transport of activated fatty acid from cytosol to mitochondria done, then the third step beta oxidation properly occurs here in the third step. This each cycle of beta oxidation liberate 2 carbon compound or 2 carbon unit, which is known as acetyl coenzyme A, which requires a sequence of different reactions here. The first reaction is oxidation here. Here acyl coenzyme A, which is undergoes dehydrogenation to form a FAD dependent flavoenzyme acyl dehydrogenase. A double bond is formed between second and third carbon here, and after that, after oxidation there is hydration occurs, where enzyme coenzyme A hydratase brings about the hydration of the double bonds to form beta hydroxy acyl coenzyme A. In this hydration, there is a formation of beta hydroxy acyl coenzyme A, which is again oxidized to form beta keto acyl coenzyme A by the help of enzyme beta hydroxy acyl coenzyme A dehydrogenase. This is the second oxidation step here, and this reaction also produce NADH here, and in this reaction there is formation of beta keto acyl coenzyme A. The fourth step is here cleavage step. Here this is a final reaction of oxidation here or beta oxidation here. There is liberation of 2 carbon fragments. First is acyl coenzyme A and second is acetyl coenzyme A. Here this reaction is proceed by the help of thiokinase enzyme, where is the cleavage of or catalyzed by beta ketoacyl coenzyme A thiolase, or it is simply known as thiolase enzyme. This is a general step which is used here for the formation of beta oxidation. Here there is acyl coenzyme A, because we are known that we are having the palmitic acid here for oxidation of this beta oxidation. So, we are using R here. So, this acyl coenzyme A which when reacted with the acyl coenzyme A dehydrogenase, and there is a formation of one high energy compound FADH2, and this FADH2 is when released from this reaction, and there is formation of trans-enoyl coenzyme A, and this trans-enoyl coenzyme A is when reacted with the enoyl coenzyme A hydrolase enzyme, and there is addition of one water molecule in this reaction, and there is formation of beta hydroxyacyl coenzyme A, and beta hydroxyacyl coenzyme A dehydrogenase enzyme reacted in the reaction to catalyze this reaction, and there is formation of one NADH molecule here, which is also a high energy bond, and there is formation of beta keto acyl coenzyme A. This beta keto acyl coenzyme A is catalyzed by beta acyl coenzyme A acetyl transferase, and there is addition of coenzyme A here, and there is formation of one acyl coenzyme A and one acetyl coenzyme A here. This reaction is repeated 7 times to form 2 molecules of acetyl coenzyme A, and this the total energies which are produced in the beta oxidation, because there are formation of various high energy molecules in this reaction. So, in beta oxidation, because in beta oxidation we are oxidize palmitic acid here. So, there is for the formation of 2 acetyl coenzyme A, the reaction is again repeated for the 7 times for the formation of 2 acetyl coenzyme A here. So, beta oxidation contains 7 cycle for the formation of 2 molecules of acetyl coenzyme A. So, there is formation of 7 FADH2, because in one reaction there is formation of 1 FADH2, there is formation of 7 NADH in 7 reactions. So, 1 FADH2 gives the 1 FADH2 gives 1 ATP. So, 7 FADH2 gives 7 ATP, and 7 NADH which is produced in the beta oxidation, which are given 14 or 27 ATP, because 1 ATP is equals 1 NADH is equal to 3 ATP here. So, there is formation of 2 FADH, 1 FADH2 and 1 NADH, and 1 FADH2 is equals to 2 ATP, and 1 NADH is equal to 3 ATP here. So, there is formation of 14 ATP here from 1 FADH2, and there is formation of 21 ATP from 7 NADH2. So, there is total formation of 35 ATP from beta oxidation proper. There is oxidation of palmitic acid in 7 constitutive repetitive reaction for the formation of these ATP here. So, 8 acetyl coenzyme A, which is oxidized in the TCA cycle or tricarboxylic acid cycle, which is also known as Krebs cycle. So, in Krebs cycles there are 96 ATPs formed. So, we also know that in palmitic acid there is use of 1 ATP here. So, there is in the activation of, in the first step, if you remember this thing, that in first step activation of fatty acid, we required 2 ATP molecules. So, we have to remove these 2 ATP molecules from here. So, there is total 129 ATPs are synthesized or produced in the beta oxidation of 1 palmitic acid, or oxidation of one beta oxidation of palmitic acid produced 129 ATPs. This is in a completed for beta oxidation of fatty acids. Now, we have to shift it towards the ketone bodies. what are ketone bodies and how they react and how they synthesized, this is in our syllabus. So, we have to know what are ketone bodies. Ketone bodies are synthesized in our body and they are the oxidized product of fat. When fat is oxidized, the ketone bodies are formed from the fat oxidation. So, ketone bodies having three different names, yeah, or ketone bodies having three different types. The first one in acetone, the second one is acetoacetate, and third one beta hydroxybutyrate. These three are known as ketone bodies, in which two are having the true ketone bodies and the third one beta hydroxy or 3 hydroxybutyrate is known as pseudo ketone bodies, because it does not have ketone group in their structure. By acetone and acetoacetate having the true keto group in its structure. So, this is known as true ketone, but beta hydroxybutyrate is known as pseudo ketone. What is ketogenesis? Formation of ketone bodies in our body is known as ketogenesis. This is occurs in the liver, and what is the definition of ketogenesis? It is a biochemical process by which ketone bodies are synthesized by breaking down of fatty acids and ketogenic amino acid. What are ketogenic amino acid? Those amino acids which are contain ketone in their structure or keto group in their structure are known as ketogenic or ketone forming amino acids. So, the enzymes for ketone bodies synthesis are located in the mitochondrial matrix, and acetyl coenzyme A, which is formed by the oxidation of fatty acid, is basically required here for the formation of ketone bodies here. So, two molecules of acetyl coenzyme A here condense to form acetoacetyl coenzyme A. This reaction is catalyzed by thiolase, which is enzyme involved in the first step of beta oxidation. So, their acetoacetate synthesis is appropriately regarded as the reversal of fatty acid oxidation. So, acetoacetate coenzyme A, when combined with the other molecules of acetyl coenzyme A to produce 3 hydroxy 3 methyl glutaryl coenzyme A, which is also known as HMG coenzyme A. This HMG coenzyme A is responsible for the formation of ketone bodies here. So, in this step, 2 molecules of acetyl coenzyme A reacted here. This acetyl coenzyme A is produced in the fatty acid oxidation, and when they react with the beta keto thiolase enzyme here, in the presence of this beta keto thiolase enzyme, and there is a removal of one coenzyme here, and there is formation of 3 hydroxy 3 methyl glutaryl coenzyme A, which is also known as HMG coenzyme A. In presence of HMG coenzyme synthase, here acyl coenzyme A and water molecule are used in the reaction for the removal of one coenzyme A to form HMG coenzyme A here. So, whenever this HMG coenzyme A is degraded here or oxidized here to form firstly acetoacetate here by the help of HMG coenzyme A lyase enzyme, and there is removal of one acetyl coenzyme A, and when this acetoacetyl coenzyme A again reacted to remove one carbon dioxide from this reaction to form acetone, and beta hydroxybutyrate dehydrogenase enzyme is also reacted with the acetoacetate to form beta hydroxybutyrate. And what is ketolysis or ketolysis? Ketolysis is the degradation of ketones to form to give the energy. So, ketone bodies are formed in the liver, but they utilized in the extrahepatic tissues. Heart muscles and renal cortex are also utilizes ketone bodies. So, these glucose is generally not utilized in when glucose is not available in our body. So, these ketone bodies are utilized here for the formation of ATPs, and for the when body required energy, these ketone bodies are utilized. Almost all tissues like intestinal mucosal cells, placenta, adipocytes and other organs which are used here, which are required ATPs for the formation of different biological function here. So, this is the ketolysis. Here beta hydroxybutyrate, which is a ketone body is when reacted with the beta hydroxybutyrate dehydrogenase enzyme to form acetoacetate. Acetoacetate is first form ketone body here by the help of one NAD to form one NADH, and this acetoacetate is when reacted with the beta ketoacyl coenzyme A transferase here. So, there is one succinate is also reacted to form succinyl coenzyme A, which is an important intermediate of Krebs cycle, and formation of acetoacetyl coenzyme A. This acetoacetyl coenzyme A again reacted with beta ketothiolase enzyme, and there is also addition of one coenzyme A in the reaction to form two molecules of acetyl coenzyme A. So, this ketolysis is reversal of ketogenesis reaction, because here is degradation or oxidation of different ketone bodies to form acetyl coenzyme A, which is the starting product, starting compound of ketogenesis. In this reaction, there is no use of or not required requirement of acetone here present, because acetone is released from the body by respiration. So, for in ketolysis there is no use of acetone, because acetone is not present in the body. Whenever acetone present in the body, it removed from the body by respiration. So, beta hydroxybutyrate and acetoacetate are utilized here for the formation of acetyl coenzyme A in this reaction. So, this is the overall reaction of ketolysis. In this chapter, in this lecture, we have completed beta oxidation of fatty acids, ketogenesis and ketolysis. These all reactions are very important for exam purpose also, and this ketolysis and ketogenesis is important for in such condition when glucose is not available for our body, and this ketone bodies are utilized in our body to form to give the energy for better performing of our body. So, this is the overall completion of this lecture. Thank you.